Hello, everybody, and welcome back to BioSC 140 Human Physiology. This video is going to cover the heart parameters, part 2. All right, so let's get right into it. The SA node sets the basic heart pattern. So we're going to look at how a heartbeat happens. We're going to look at the heartbeat cycle, the heart cycle, from a few different viewpoints, and then we're going to put them together. So we're going to look at the cycle as a whole a few different times in a few different ways, and then we're going to compare them. This should give you a good understanding of what's happening at each moment throughout a cardiac cycle, throughout a heartbeat. So SA node sets the basic heart pattern. We talked about that in part 1. If you need to review, please go back and review. The SA node spontaneously depolarizes. It doesn't need outside stimulus in order to start the muscle action potential, to start the muscle contraction. Outside influences do affect it, but it does not require outside influences. Because of why, here's a good question. Why does that action potential, or how does that action potential spread from one cardiac muscle cell to the next? How does that contraction, how does that muscle action potential spread from one cell to the next? It spreads through direct cytoplasmic connections called gap junctions, which are found in the intercalated discs in between cardiac muscle cells. So depolarization spreads through the atria to the AV node. So a little more review. As that depolarization event spreads from one atria to the other atria and through the atria, notice how it kind of starts at the top and spreads down. This is the analogy that I had with my wife getting toothpaste out of the toothpaste tube, where she just kind of squeezes it randomly and it's not very efficient. And I start at the bottom and I kind of squeeze from the bottom upward, and it's a more efficient way to get the toothpaste out. That's what it's doing here. It's the contraction starting at the top and it's spreading down, and it's making the atria push that blood into the ventricles. So what prevents the action potential, this electrical event, from spreading from the atria to the ventricles just freely? There's a reason why this action potential spreads throughout the atria, but then kind of stops and doesn't spread to the ventricle. The atrioventricular septa. The atrioventricular septa does not allow this action potential to pass directly to the ventricles. So this electrical potential, this electrical event, this contraction event, it starts at the top, works its way down through both atria, and then it gets to the AV node right here. At the AV node, the conduction is slowed, and it travels through something called the bundle of His, and then the left and right bundle branches to the apex of the heart. Remember, the apex of the heart is in like the bottom, like if you're, for me, like with relation to my heart, the bottom left. So it's like this point down here is the apex. This point down here is the apex. So it spreads throughout the atria, gets to the AV node, goes through the bundle of His, and then the left and right bundle branches to the apex. And from the apex, it goes into something called the Purkinje fibers. That's a vocab word. I do want you to know the Purkinje fibers. And so it basically, contraction starts at the top, spreads through the atria, pushes the blood down to the ventricles. It then skips through the ventricles to the apex, and then starts that contraction from the apex, kind of moving upwards. And this allows, again, for the efficient contraction of the heart. The ventricles up here, I mean, the ventricles are pushing blood into, these tubes up here. And so it's contracting in that manner to push up into the aorta and up into the pulmonary artery. I want you to know this order of operations. I have asked questions before about this order of operations. You should know how this electrical event flows through the heart. So yeah, as each part depolarizes, you get the contractions. So contractions of the atria, it travels from the AV to the bundle of His, to the left and right bundle branches, to the apex, then out across the Purkinje fibers, contracting the ventricle. That's one way of looking at the heart, one manner of looking at the heart throughout the cardiac cycle. Is kind of the path of the depolarization event. We're now going to look at the cardiac cycle via the ECG or EKG electrocardiogram. You'll see it both ways, ECG and EKG. Some languages spell cardio with a K. So ECG is electrocardiogram, EKG is electrocardiogram also. It kind of reminds me of vitamin K. So vitamin K is a clotting vitamin, and the scientist that discovered it, I think he was like Austrian or somewhere in Eastern Europe, and his language, they use K to spell clotting. So vitamin K is vitamin clotting. Fun fact. All right, so ECG is considered non-invasive. What does it mean by non-invasive? It means no needles. You're not poking the skin at all. You just put some electrodes on top of the person's body. It measures the summed electrical events or activity of all cardiac cells. And it does this via volume conductance. It's underlined, it's a vocab word, make a flashcard for it. Electrical activity passes through your blood. There are electrolytes in your blood. What we're able to do is we're able to put sensors on your wrists and your feet or your ankle, and we can actually detect the electrical activity of your heart by putting sensors on those parts of your body because of volume conductance, because that electrical event travels through our bloodstream. All right, so a few things about the ECG. It's very diagnostic. I'm actually going to skip ahead a slide. This is what the ECG looks like. We've all seen it in movies. The person's lying in the hospital bed, and you have the screen next to him, and it goes beep, beep, beep. That's what this is. So we're going to learn all about it. So it's very diagnostic of a few things. It lets you know the heart rate. So you have a very accurate measure of heart rate using an ECG. And like when you take someone's pulse and you measure their heart rate that way, you have to wait for 15 seconds, wait for 30 seconds, and do some math. The ECG can give you you don't need to wait 15 seconds. It's like it can tell the exact amount of time between one heartbeat and the next, and tell you if that was extrapolated over a minute, what the heart rate would be. So it's a very short time frame, a very accurate picture of what that heart rate is at that moment. It can tell you about previous myocardial infarctions. What's a myocardial infarction? We haven't gone over this yet, but that is the medical term for the heart attack. Cardiac abnormalities, conduction abnormalities. So conduction is referring to kind of that depolarization pathway that we talked about in the last little bit that we just finished talking about. So any abnormalities in that. And it can even tell you about the different heart chamber, the size of different heart chambers, which does have some healthcare implications. It can help you know what's going on with the patient and give better care in certain situations if you know about the heart chamber sizes. So it tells us a lot of information. ECG consists of a series of waves, segments, and intervals. These are vocab words. You're going to see it in lab. It's here in lecture. You're probably going to see it on the lab quiz, and you're probably going to see it on the lecture exam. Nudge, nudge, wink, wink, hint, hint. Make sure you know these. So a wave is a deflection above or below baseline. So we have our baseline, which is this flat line right here, kind of this flat part, this flat part. A deflection above. So right here we have the P wave. It just kind of goes up. That's it. There's not a lot going on. So, there's a lot going on, but it's just a simple little deflection above or below. Cool. A segment is the baseline between waves. The baseline between waves. So right here we have the P wave, and then we have the QRS complex. In between those is the PR segment. It's the baseline between waves. And the interval is a combination of waves and segments. So we have the QT interval, we have the PR interval. So wave, deflection above or below baseline, segment is the baseline between waves, and an interval is the combination of waves and segments. So this is what normal looks like. What is going on in the heart at each moment? That's what I want you to know. What is going on in the heart at each moment throughout this ECG? What's going on during the PR interval? What's going on during the P wave? What's going on in the QT interval? What's going on during the T wave? That's the knowledge I want you to have, and that's the type of question you can expect on the examination. So the ECG activity corresponds to electrical events in the cardiac cycle. So right here, we can kind of see how we're starting to overlap different ways of looking at the cardiac cycle. We just went over kind of the conduction. Remember this kind of graph right here. And we're going to have our ECG right next to it. So you can see what part of the ECG is happening at that corresponding conductance event. So the P wave stands for atrial depolarization. The P wave starts when the SA node depolarizes and you start to get depolarization in the atria. So P wave starts, contraction of the atria, or depolarization of the atria. Right here, QRS complex. The QRS complex, two things happen during it. There's always every semester, people miss this question on the exam. Every time I ask it, I've asked it in different ways. Two things happen during the QRS complex. Ventricular depolarization, so ventricular contraction, and atrial repolarization, so atrial relaxation. So right here, we get the start of the Q wave, and then the R wave, and the S, the QRS complex right here. You can see how we go from contracting atria to relaxed atria, so we get atrial relaxation. And you can also see how the ventricle starts to contract. So two things happen during the QRS complex. They're both kind of hidden in there at the same time. Ventricular depolarization, atrial repolarization. The T wave is when the ventricle repolarizes. So once we start to get the T wave up here, we start to get some relaxation. So T wave relaxation within the ventricle. The ECG estimates duration of systole and diastole. So where systole is contraction, diastole is relaxation. You can break systole and diastole up into atrial systole, so atrial contraction, and ventricular systole, or ventricular contraction. And you can do the same for diastole, atrial diastole, atrial ventricular diastole, so ventricular atrial relaxation. So P wave occurs. P wave occurs, we start to get contraction or depolarization in the atria. Start to get contraction in the atria. It's contracting until the QRS complex. At the QRS complex, the atria relaxes and the ventricle, so that's atrial systole. Atria starts to contract, P wave, it relaxes QRS. Atrial systole is this segment right here. Ventricular systole starts at the QRS complex. Ventricular systole starts at the QRS complex. And it ends with ventricular relaxation at the end of the T wave. Ventricular systole is the QT interval. Starts at Q, ends at T. QT interval is ventricular systole. The TQ interval is ventricular diastole, ventricular relaxation. It starts at the T wave. It ends at Q. So T all the way back around to Q. I do want you to know about the QT interval and the TQ interval. Excuse me. All right, we're going to look at the heart cycle in another way now. All right, so the heart contracts and relaxes in a rhythmic cycle. So we have atrial and ventricular diastole. Remember, blood flows from high pressure to low pressure. What is the normal path of blood? This is a question. What is the normal path of blood through our heart? So, venous system to right atria to right ventricle to pulmonary artery, pulmonary capillaries, pulmonary vein, back to the left atria to left ventricle to our aorta. We have 4 valves within our heart. We have valves in between our atria and our ventricles, and we have valves between our ventricles and the corresponding artery. If pressure is ever set up so that blood will flow in the direction we want it to flow, the valves are going to be open. If pressure is ever set up across the valve so that blood is going to flow not in the direction we want it to flow, that valve is going to close to prevent it. Valves prevent backwards flow of blood. They ensure one-way flow of blood. So, atrial ventricular diastole, pressure is already set up so that blood is going to flow from our veins to our atria to our ventricles. Remember, 2/3 of our ventricular filling happens passively. Then we're going to get atrial systole. So that last little bit of filling for our ventricles, we need a little extra help in order to get that extra little bit in and get it completely full. So the atrias contract. We have atrial diastole, which pushes that last 1/3 of blood into our ventricles. We then have atrial diastole, so the atria relaxes, and the ventricle contracts. The ventricle contracts. When the ventricle contracts, it's going to push blood from our ventricle into our arteries. Ventricle into our arteries. Now, also while that's happening, blood is passively flowing from the veins of the atria. So diastole, relaxation, systole, contraction. So right here, atrial systole, ventricular diastole. So let's start right here. So 1, atrial and ventricular diastole. So blood is passively flowing from veins to atria to ventricle. That's about 0.4 seconds in this example. 2, atrial systole and ventricular diastole. So atria contracts, pushes blood into the ventricles, completes the filling of the ventricles. We then have ventricular systole and atrial diastole. Ventricles contract, pushing blood into the corresponding arteries. We're going to go into a little more detail about this cycle. pay attention to pressure, pay attention to when different valves are open and when they are closed. Remember, the general rule of thumb, this actually is just how it works. If pressure is set up so that blood is going to flow the direction that you want it to flow across where a valve is, the valve is going to be open. If pressure is set up so that blood is not going to flow in the direction you want it to flow, the valve closes instantly. So, all right, so the heart contracts and relaxes throughout the cardiac cycle. So, ventricular diastole and atrial diastole. This is when the heart is relaxed. We're going to have the lowest pressure in the ventricle, a little bit higher in the atria, a little bit higher in the veins. So blood is flowing from veins into the atria, into the ventricles. This is when the system's already set up, the pressures are already set up so that passive flow is occurring in the ventricles. 2/3 of the ventricular filling passive during this. A majority of the cardiac cycle is spent here. At rest, about 2/3 of the cardiac cycle is spent in this phase. It changes some during exercise. During exercise, it might be 50%, but during rest, it's about 2/3 of the cardiac cycle is spent at this phase relaxed. So we have 2 sets of valves. Well, there's 4 valves, but there's 2 in between atria and ventricles and 2 between the ventricles and the corresponding arteries. So blood is flowing from the atria to the ventricle. We know that pressure gradient is set up in a favorable way. So the valve, the atrioventricular valves, the valve between the atria and the ventricle, is going to be open during this time. We know that the pressure in the ventricle is going to be very, very low because blood's flowing into it. The corresponding arteries tend to be, pretty high. The aorta fluctuates between 120 and 80. That's high. So the semilunar valves, the valves between the arteries and the left ventricle, are going to be closed. So during this phase, AV, atrioventricular valve, open, semilunar valve between the ventricle and the corresponding artery, closed. We're then going to get atrial systole. So we have 2/3 of the blood passively flow into the ventricles, and there's going to be a point where that ventricle starts to get a little tight, starts to get a little full of blood, right? And it starts to increase the resistance, increase the pressure within the ventricle. And so in order to get a complete filling, we need a little extra kick. We need that atrial kick. We need the atria to contract to push that extra little bit of blood into the ventricle. Atrial systole, atrial contraction. Now, the pressure is going to be higher in the atria than in the ventricles because the atria is contracting. AV valve is open. AV valve is open. There's still very low pressure in the ventricle, relatively higher in the corresponding artery. We need to prevent backflow. We need to prevent flow of blood from the artery back to the ventricle. So the semilunar valve is going to remain closed. Semilunar valve closed. Vocab term. You need to know this one. It's going to come up a few different times. End diastolic volume, or EDV. Volume of blood in the ventricles at the end of diastole. So at the end of the relaxation phase, ventricular relaxation phase, when the ventricle is completely full, how much blood is in there? It's composed of the passive flow, that first 2/3 of passive flow. It's composed of the active flow. So how much does the atria kind of push in with its contraction, and the remainder from the previous cycle. Not 100% of blood is pumped out. In a normal situation, a high percentage is pumped out, but not 100%. So the remainder from the previous cycle. Assume average adult human, 100 milliliters. Average adult human, 100 milliliters is the end diastolic volume. A very athletic person, like Luke Skywalker, it might get up to 135 milliliters. 135. So atrial systole, the contraction of the atria to push that last 1/3 of blood into the ventricles to get them completely full. We're then going to move into isovolumetric systole. Isovolumetric ventricular systole. Yes, there's going to be multiple parts of ventricular systole. So isovolumetric, let's break the word down. Iso means same, volumetric, same volume, ventricular systole. So we're going to start to have contraction of the ventricle. And we're going to have no change in volume, but we're going to have an increase in pressure during this isovolumetric phase. So ventricle is completely full of blood. It's time to contract the ventricle. So the ventricle starts to contract. When the ventricle starts to contract, the pressure in the ventricle is going to become higher than the pressure in the atria. Do we want blood to flow from the ventricle to the atria? We do not want blood to flow from the ventricle to the atria. Pressure is set up so that we have higher pressure in the ventricle, lower pressure in the atria. If we don't stop the blood from flowing that direction, it's going to want to flow that direction. So during isovolumetric systole, the AV valve closes. Remember, the AV valve was open during ventricular diastole. And now that we're entering ventricular systole, the valve closes. And it closes at the moment pressure in the ventricle gets higher than the pressure in the atria. The moment pressure gets higher in the ventricle than the pressure in the atria, that valve, that AV valve is going to close. Now, it's going to take some time to go from basically 0 pressure up to 80 pressure or a high pressure in order to get the blood into the artery. There's a period of time when the pressure in the ventricle is higher than the pressure in the atria, so the AV valve is closed, and the pressure in the corresponding artery is higher than the pressure in the ventricle. Remember, things flow from high pressure to low pressure. The pressure is higher in the artery than it is in the ventricle, so blood wants to flow back into the ventricle. We don't want that. Semilunar valve remains closed. Remember, semilunar valve was closed in atrial, in ventricular diastole. Now here in isovolumetric systole, semilunar valve is closed. Highest pressure in the artery, middle pressure in the ventricle because it's contracting, it's generating some pressure. Lowest pressure in the atria, in the atria. All valves are closed. All 4 valves are closed. AV valves are closed, semilunar valves are closed. Isovolumetric, all the valves are closed. There's no blood leaving or entering the ventricles. Isovolumetric, it's staying the same size. What's changing is the pressure. The pressure is increasing in the ventricles as the heart is contracting. That's what's changing, is the pressure in the ventricles, not the volume. First heart sound. So when people describe the sounds of the heart, they go lubbed up, lubbed up. When you listen to the heart, there's kind of two sounds to it. Lubbed up, lubbed up. That first sound you hear, basically the sounds are valves closing. So you hear the AV valves closing, that's the first heart sound. The first heart sound is when the AV valve closes. AV valve was open during these 2 phases. Right here, as the ventricle starts to contract, AV valve closes. You hear the valve slam shut. Slamming doors are loud, right? Slamming the AV valve shut, that's your first heart sound, the lub-dub. Isovolumetric contraction is a very brief period. It's a very brief period. At the moment, remember, we're generating pressure during isovolumetric. At the moment, the pressure in the ventricle is higher than the pressure in the corresponding artery. We're going to have the pressure gradient set up in such a way that blood's going to flow the direction we want it to, right? So the moment the pressure in the ventricle is higher than the pressure in the corresponding artery, isovolumetric contraction is going to end, and ventricular ejection is going to start. The semilunar valve is going to open the moment the pressure in the ventricle is higher than the pressure in the artery. The moment the pressure gradient is favorable for blood to flow the direction we want it to flow, the semilunar valve is going to open, blood is going to flow from the ventricle into the artery. Semilunar valve opens. This gives us our systolic blood pressure. So we're going to be pumping up that artery, pushing blood into that artery at this moment. We still have closed AV valves because we have high pressure in our ventricle, low pressure in the atria. Pressure in the ventricle is higher than the pressure in the corresponding artery. We have the semilunar valves open. Blood is being pumped through the semilunar valve into the corresponding artery. Once a large portion of the blood has been pumped out of the ventricles, once the contraction phase is all done, we're going to enter isovolumetric diastole. So, ventricle is done contracting, it's starting to relax. The moment the pressure in the corresponding artery is higher than the pressure in the ventricle, we don't want blood to flow backwards, the semilunar valve is going to close. Now, during isovolumetric, we're not changing volume, we're changing pressure. There's still higher pressure in the ventricle than there is in the corresponding atria. So AV valve closed, semilunar valve closed. The closing of the semilunar valve here is our second heart sound. Remember, heart sounds are closing valves. The first one is closing AV valve. The second one is closing semilunar valve right here at the beginning of isovolumetric ventricular diastole. End systolic volume, ESV, end systolic volume is the volume of blood in the ventricle at the end of systole. So, which is going to be a larger volume? End diastolic volume or end systolic volume? What's going to be a larger volume? End diastolic volume or end systolic volume? End diastolic volume is how much volume is in the ventricle at the end of the relaxation phase, at the end of filling. So end diastolic volume is generally larger than end systolic volume, which is the volume after all the blood's been pumped out. Students get those two mixed up. Put stars next to those two. Students get those two mixed up all the time. So here's your heads up, here's your warning. Make sure you know the differences between those two. For some reason, know that end diastolic volume is larger than end systolic volume. End systolic volume, don't get those two mixed up. It's common. All right, and so then we have isovolumetric ventricular diastole, perfect. It's about 30 milliliters. Remember, there was a little bit left over in the ventricles after the contraction. It's about 30 milliliters to the average person. And systolic volume. And then we get back to atrial and ventricular diastole. Know those 5 phases. Atrial and ventricular diastole, blood is passively flowing into the ventricles. Know what valves are open when, know what valves are closed when, know what's going on with pressures and have blood flow, and know this cycle. All right, now let's put it all together. So we just looked at kind of this cardiac cycle in a number of different ways. So we looked at it with conduction. We looked at how the depolarization event passes through the heart. We looked at it with the ECG. We looked at it with the flow of blood and contraction of muscles, opening and closing of valves. Here is kind of the summary chart where we see it all close to it, all together. I want you to pay attention to these two charts in the middle. So look at this one right here. So this one is going to show you the pressure in the different chambers at each moment. So right here, we're starting at ventricular and atrial diastole. Pressure's pretty high in the aorta. Remember, pressure in the aorta fluctuates between, 120 and 80. Pretty high. Pressure in the ventricle, this is left ventricle, almost zero. Pressure in left atria, slightly above. At this point, where is blood? Is blood flowing? At this point, blood is flowing from the left atria into the left ventricle. This is that 2/3 passive flow that I was talking about. Pressure is a little bit higher in the atria, so it goes from high pressure to low pressure in the ventricle. At some point, we're going to get that left atria to contract to pump that last 1/3 of blood into the ventricle. So you can see the pressure in the atria goes up a little bit, pressure in the ventricle goes up a little bit, but pressure is still higher in the left atria. We get the left ventricle full of blood, and then we get the ventricular contraction, left ventricular contraction. Right when the left ventricle begins to contract, the pressure in the ventricle is going to become higher than the pressure in the atria. And at that moment, where this red line crosses the blue line, the AV valve closes to prevent backflow of blood. AV valve closes right when that pressure, right when that red line crosses the blue line. AV valve closes, we get our first heart sound. Isovolumetric systole is going to occur in between this line right here and this line right here. When the red line, the pressure in the left ventricle, is in between the pressure in the left atria and the pressure in the aorta. In between, when the red line is in between the black line and the blue line, that's isovolumetric systole, isovolumetric contraction. We're increasing pressure, we're not changing volume. At the moment this red line crosses the black line, at the moment the pressure in the left ventricle gets to be higher than the pressure in the aorta, the corresponding artery, semilunar valve opens. And we get flow from the ventricle into the aorta, pumps it up to 120 millimeters of mercury. At the moment the ventricle starts to relax and the pressure in the ventricle gets lower than the pressure in the aorta, that's the moment the red line crosses back underneath the black line. That's when the semilunar valve closes to prevent backflow into the ventricle. We get our second heart sound, and we begin isovolumetric diastole. Isovolumetric diastole is in between this vertical line and this vertical line. And at the moment this red line goes below this blue line, we get the end of isovolumetric diastolic relaxation. And the AV valve is going to open, allowing blood to flow from the atria back into the ventricle. This one down here shows you the volume in the left ventricle during each phase. All right. If you have any questions at all, please let me know. I'm here to help out. I'll see you in the next video, everybody.